Well, good morning, everyone. For micro, it's not very many. Although, on that today, I mean, part of it is my lectures are freaking lame and you got the PowerPoints. But this is not like my class. I'm sort of the interim instructor here. Regardless, we're going to hand back exams. Mm-hmm. So I'm going to call out everybody's name and then...
It was like Ferris Bueller.
Yes, it is.
Bueller?
Bueller? Anyone? All right. Taylor?
Oh my gosh.
I'm sorry you're not feeling well.
No.
Let's see. Angel, how are you? He's in St. Louis, I think.
Thank you.
You're welcome. No problem. Very good. Uh-oh. Just for the audience, the Caitlin's glaring at me. But no, let's go through it a bit here. We can negotiate some answers. Let me get one to go off of.
Did
All
you
right.
grade the short answer this time?
Kind of. I looked over them, and I was happy with them.
Gave you good enough?
Yeah.
Okay.
Yeah,
Part of it-
number 22, I was having.
Is that I get, I grade it and I'm like, this is about where I want the grades to be. I don't want to take any more points off anybody else. So I look over them and go, yeah, we'll go through it a little
Let's
bit.
go through them.
Okay, so bacteria and archaebacteria, they are fast reproducers. So they are asexual reproduction because you don't have to have a partner. And mutations are how they generate genetic variety. Many eukaryotes reproduce slowly, and sexual reproduction is the way that many eukaryotes are actually operating. Because not all of them, though, because yeast don't, but many fungi do. Anyway, regardless. Okay, so then we have the log phase of bacterial growth is where the cells are dividing very rapidly because they are particularly happy, lots of nutrients, there's not a lot of toxic metabolites, i.e. they haven't peed much in the pool. On the other hand, during the decline phase, they start to die off because the nutrients are limiting and they generate all these different kinds of toxic metabolites that are not good for them or for anybody else. Animals, we require aerobic respiration. We are therefore obligate aerobes. We can carry out fermentation a bit, but not for very long, and it doesn't do you a whole lot of good. Then there are some bacteria that are obligate anaerobes, very toxic, like botulism. And again, it's just one of the things that this, we touched on here in this exam of, there are things that bacteria do that we just don't even begin to do, using anaerobic respiration being one of them. All right, let's see. Question 4 is a question about enzymes, and we've got competitive inhibitors in which the inhibitor is going to bind the enzyme active site and block its action. Non-competitive, we actually are generating a site on the enzyme that causes a change in shape and stops its action. You could add more substrate, but a non-competitive inhibitor, you're never going to be able to reach the maximum velocity. Let's see. So enzyme pathways, lots of different chemicals. Glucose is used to produce ATP. During respiration, let's see, but glucose and Krebs cycle intermediates produce monomers such as amino acids to make proteins. Okay, then question 6 about the control. High temperatures and many disinfectants are often denaturing proteins, such as enzymes, and then disrupting membranes, which causes cells to rupture. Question 7 is the phenol coefficient. And let's see, if a chemical receives a score greater than 1, it's considered to be fairly toxic, classified as a disinfectant. And you may have been right, Pearson, that disinfectant may not be on the word list, but
Yeah, I was a little confused on
Yeah.
that, so I just
Sorry
couldn't
about that, I
answer
missed it. So, yeah, you put a little note, which I counted, I was taking disinfectant or antiseptic.
Okay.
Antiseptics are what we talk about as things that are slightly less toxic and you can use them on your skin. So when we're spraying down the tables, we're spraying them down with disinfectants, which is consequently why they say don't get this on your skin. Let's see, essential genes are on the bacterial chromosome, and then the optional genes are on plasmids. And these are things that can be transferred around back and forth too. Let's see. Though often inefficient, cells can import DNA directly from their environment. Transformation is taking it up with a plasmid. The DNA transfer with a virus is transduction, and DNA transfer between two different bacteria is a conjugation, and that requires the F plasmid and stuff. So transposons are DNA sequences that are able to move from one DNA to another, and they are responsible for bringing in different kinds of antibiotic resistance genes. Yeah. All right, let's see. So the multiple choice questions, here we go. Some of these I think are fairly obvious, but some not so much. All right, in the decline phase, the death rate exceeds the reproductive rate, and so we're having the number is dramatically decreasing. That's A. Psychrophiles. A number of people got this wrong. I actually looked it up on the Googles, as perhaps many of you did as well, and it does say in deep ocean trenches. That's because deep ocean trenches are cold. A barophile would be a better term for somebody growing in a deep ocean trench. Psychrophiles specifically are referring to those guys that grow in colder temperatures. And so when you look at, it's like the polar regions, and the best notion would be in a refrigerator.
The reason why I didn't pick a refrigerator is because I didn't know if it was cold enough.
Oh, well, psychrophiles are those guys that can grow from like 10 degrees, which is about 40 Fahrenheit,
Okay.
down to minus 20 or so, which
Yeah.
is about 0 Fahrenheit. But yeah, fair enough. If in the end, just telling this to all the TV audience, if your grade depends on 2 points, hey, I give
Yeah, I
you
was
2
just
points.
curious,
Like your overall grade. So anyway, yeah, okay. So getting electrons from inorganic material, carbon from CO2, and energy from sunlight. First off, if you're getting energy from sunlight, you're going to want a photo something or other. Using inorganic materials, these are lithotrophs, and carbon from CO2 is not meaning you don't have to take in things like glucose or other organic molecules. C is the best answer. You're a photolithoautotroph. And barophiles are the guys that are going to be found in the deep ocean. High pressure, very weird organisms there. Let's see, facultative bacteria, they can grow with or without oxygen. If you ask them, they probably would prefer oxygen. They can grow a lot faster, but they will operate without oxygen. All right, let's see. Question 6 was a little bit of an interesting one, in that our stomach would not be able to protect us from pathogens that are also, the best answer would be acidophiles, in that acidophilic bacteria, like Helicobacter pylori, they actually can operate in a very acidic environment, and in fact they do it because they're dumping out with urease. They're kind of changing their local pH, but they've adapted to growing in a very acidic environment, and so the stomach acids can't protect you from those kinds of guys. They're not going to protect you from acidophiles. Let's see. So catabolism. This is the breakdown of stuff, going from the more complex to the simpler, and so pretty much everything here is anabolic, that is, making proteins, making large things that are larger, making nucleic acids from nucleotides, and the best answer is fermentation, is an example. Let's see. Enzymes are essential for life. The way enzymes work is they're reducing the activation energy. So A. Feedback inhibition is what we're using when there is too much product from an enzymatic reaction and we begin to have the enzymes start to slow up. let's see, instead of using oxygen to remove electrons, anaerobic respiration is most likely to use something like, the best answer is probably going to be some of the ions, things like iron, nitrate, or sulfate. These other things are not standard places to dump electrons onto. If it was NAD, that might be something, but NADH is a reduced form of it. It can't pick up another electron. Let's see, instead of using glucose for chemical energy, lithotrophs are likely to use something like, again, these different ions. So that's probably the best answer. Yeah. All right. Facultative anaerobes, they prefer to use aerobic respiration when possible because you get a lot more ATP. Remember, when we talk about with glucose, in the presence of oxygen, one glucose is worth 34, 36 ATP. One glucose in fermentation is worth 2 ATP. So you have to eat a lot of sugar to generate the equivalent amount of ATP. Let's see, killing pathogens with heat but not sterilizing to prevent damaging the food. That is our friend Louis Pasteur's technology of pasteurization. UV irradiation is going to be pretty good at doing solid surfaces. Liquids, you can't get the UV light through it very well. Packaged foods, it's like, you covered them up, so that's not going to work. You could sterilize the surface, fine. Heat-sensitive foods, again, you don't get good penetration. And skin surfaces is a great way to give yourself a bad sunburn. Salt, sugar, freezing, and drying. The best answer here is removing cells liquid water. The net result from that is going to be denaturing proteins and disrupting membranes and destroying the cell wall, probably not. But the initial thing that is happening or it's causing it all, is removing the cell's liquid water. Filtration does not do a very good job of removing viruses. Let's see. Unlike disinfectants, antiseptics, you can treat cuts and wounds. Sterilizing surfaces, they would work, be used in foods, various things like that. The best answer is probably treat cuts and wounds. Main goal of these things is to reduce the number of the pathogens on the surface. Let's see. Unlike bacteria and our archaebacteria, the eukaryote chromosome is linear. The R factor is most likely to carry drug resistance genes from one bacterium to another in the process of conjugation, I think is the best answer there. Let's see, which the following cannot produce or enhance genetic variety in bacteria. Mutations do a good job, and then anything where you bring in DNA, which is transduction, conjugation, and transformation, that's going to be ways of enhancing genetic diversity. Just straight cell division does not do a very good job of it. Let's see, responsible for any new trait, and that's going to be mutations. One of the points we were trying to drive home here was mutations occur randomly, and then you select for these mutations if they're doing something interesting for you. Transposons are hopping from one DNA to another, and they're able to take drug resistance genes and bring them into different chromosomes. Let's see, eukaryotes, we can enhance genetic diversity through the process of sexual reproduction. So you bring in a different set of chromosomes and scramble them around. Let's see, eukaryotic chromosomes are linear. They're not compact and small. They're are generally not one per cell. They're in the sex cells, like the gametes, they would be, but otherwise there's two. Have only optional and handy but not essential genes? No. One origin of replication, a rather nerd point, but no. Again, multiple origins of replication. Let's see, horizontal gene transfer is where we're actually transferring DNA from one cell to another rather than asexual reproduction. And that makes D the best answer. Let's see, 27, not possible according to the RNA, or to the central dogma of biology. We know that DNA is responsible for directing its own replication, yes. RNA carries information used to make proteins, yes. Proteins reflect DNA sequence. They do. That actually is a pretty good answer there, in that while you can't have necessarily the exact nucleotide sequence of a gene, you can have a pretty good guess. But regardless, the DNA sequence is reflective of the amino acid sequence instead of course the other way around. Let's see, mutations occur randomly. This is, in fact, one of the central points. D is the best answer, that environmental stress, like antibiotics, causes genetic mutation. And environmental stresses are selecting for existing mutations. There are a whole range of things that can cause mutations, but the mutations are random. And so then you have to have the selective pressure to actually have them become part of the population. Let's see, conjugation. The best answer is E. DNA passes from an F plus to an F minus bug, and not the other way around. And to pass chromosomal DNA from one bacterium to another, you need an F plus strain. For transformation to occur, this is where we take plasmids and put them into bacteria cells, and this is the experiment of Oswald Avery and MacLeod. The cells are said to be competent. All right, so let's see. And growth curve, the importance of the stationary phase. This is where the bacteria are operating and stabilizing and adapting to the environmental conditions. And we've got the preparation then for all kinds of different opportunities in the production of different kinds of materials in there. Pathogens are mesophiles and neutrophiles, but never barophiles or halophiles. Well, if they're going to be pathogenic to you and I, they need to be able to operate within our conditions, which are fairly moderate. The saline, about 0.9% saline, like phosphate buffered saline or physiological saline, a pH of around 7.2 or so. Let's see, feedback inhibition is critical because this is how enzymes are able to generate just exactly the right amount of product and not make too much. It's also how you can, if the product gets too low, you can relieve the inhibition and start making fresh product. Enzymes and substrates interact by the induced fit. This is the idea of how we can have the lowering of activation energy by slightly changing the shape of the substrate such that it's now more sensitive to hydrolytic attack and things like that. Let's see, advantages and disadvantages of sterilizing with heat. Heat is a really good way to sterilize stuff. Things die off pretty easily. It's cheap, but you also can end up damaging materials, denaturing proteins, things like that. Using an antiseptic, not disinfectant. So disinfectants, as we say, just as a term, are much harsher and they're going to be actually quite damaging. So there's all kinds of disinfectants that work really well, but you can't put them on your skin. Plasmids and transposons, these are DNAs that can move between cells and then can carry other genes with them. Mutations in antibiotic resistance. This is interesting, you could write a whole thesis on this, is that mutations are responsible for antibiotic resistance, but it is only when you begin using antibiotics you select for those specific mutations. Poor medical practices created the drug resistance problem. Essentially, excessive use of antibiotics is a way of doing that because you then begin selecting for those resistant bacteria. Vertical versus horizontal gene transfer. Vertical gene transfer is when you have parents dividing into two, or the parent, to produce the offspring, if you would. And then horizontal gene transfer is when you have two individuals transferring DNA back and forth. All right.
What was the answer for 19 and 20?
19 and 20. Let me see. Unlike bacteria and archaea, eukaryotic chromosomes are linear. That's 19. And 20 is the R factor is being used in conjugation. All right. Okay, so let's see. Lab-wise, we had some interesting issues with our Street plates, most folks didn't have really good street plates, and because of the way the holiday laid out, it was hard to kind of recover from that. I think what we're going to do is probably magically say everybody's street plate worked, and everybody's going to get a plate of their specific bacteria strain that we can then begin doing metabolic tests with. And we'll tell you, yes?
Did my redos work well?
I can't remember. I came in and pulled them out on Friday, but I can't remember what they look like. The MSA never really did anything, so there's that.
I never get results, so.
Oh,
Taylor and I never did.
yeah, she was doing some interesting staining. But we'll get some results next time. And you did get some stuff with the glucose, with the different fermentation, things that turn from red to yellow.
Oh.
If you looked at them, maybe. We'll be working on it. And so we're going to be doing some different, some interesting tests and things using monitoring growth of bacteria this week, and start working a little bit more on our unknowns and kind of go through how to do that. We're set for another exam that's going to cover parts of the virus stuff, but not all of the virus stuff that we've been covering, and a lot of the bacterial, maybe actually mostly just the bacterial stuff, the upper respiratory, lower respiratory, GI, and we're probably going to drop that again on Friday.
This Friday?
Yep.
Oh my gosh.
Why? You know what that?
No, I have so many tests in the next 3 weeks. In one class, I have 3 tests back to back.
That sucks.
It does, and the one I have next week, I have to get 100% on.
I'm sure for you, but you had time to work on this.
No, I'm not stressed about that. It's
Good.
just
I know.
the second half of the semesters are